Welcome to PSN Bio Academy. If you are new to my channel, this is Fazna Imran, and this is the place where you can top up your biology knowledge with our past paper explanation through very short and clear video series. Currently, we are focusing on PSN Edexcel International Advanced Level, paper reference WBI 1401, Unit 4, Energy, Environment, Microbiology and Immunology.
Today we are going to talk about chromatography. Now my first question is, why do we need to study chromatography? Yes, it has many applications for scientists, lab personnel and researchers. But what about you? As an IAL biology student, most students often feel that chromatography isn't very important or relevant to their exams. So let me clarify that with two main reasons why you must learn the steps and process of chromatography. Reason 1, to understand the core practicals. If you want to answer the core practical questions effectively, you need to understand how chromatography works. Without knowing the process, you will struggle to apply it in exam scenario. And reason number 2, it's tested in actual exam questions. For example, in the June 2023 paper, there was a question where you were given a diagram of a chromatogram showing antibiotics, not chlorophyll pigments. And the question asked, describe how this chromatogram could be produced. And this proves that chromatography isn't just a theory topic, it's actively assessed even in recent papers. So it's not something from the distant past, it's relevant right now. And in the future, they might ask for the procedure using chlorophyll pigments as well. With that in mind, let's now dive into the steps of chromatography.
So, what's chromatography? First, let's understand what chromatography actually means. It's basically a lab technique used to separate and analyze the components of a mixture based on their different physical and chemical properties. In simple words, chromatography is a separation technique, just like how we sometimes do decanting to separate a precipitate from a supernatant. Chromatography works in a similar way, but with more precision, and it helps us break down mixtures into their individual parts. Let's talk about the phases of chromatography. So, what are the two phases involved? In every type of chromatography, you will find two main phases, the stationary phase and the mobile phase. The stationary phase is the one that stays fixed in place, while the mobile phase is the one that moves through it. So these two phases work together to separate the components of a mixture based on how they interact with each phase. Now it's time to look at the different techniques used in chromatography. Remember, chromatography is a broad topic, and under these, there are several types of techniques. Let's go through a few of them. First, we have column chromatography. In this method, we use a column filled with a stationary phase and different solvents move through it. Based on how they interact with the phases, they get separated. Next is paper chromatography, which is the one we actually study in our syllabus. Here we use a piece of paper as the stationary phase and a liquid solvent acts as a mobile phase to separate the components. Then comes gel filtration chromatography. In this technique, a gel-filled column is used to separate substances, usually based on their size. And finally, we have gas chromatography. This is a more advanced and modern technique used in high-end labs. It separates and analyzes components using gases instead of liquids. Now, if you have looked at your past paper questions under the topic of photosynthesis, you'll notice that one common task is to separate chloroplast pigments using paper chromatography. So in this part of the video, let's walk through how to actually carry out chromatography to separate the pigments present in a leaf. Let's get started with the practical steps. Let's start with the materials you will need for this practical. First, you will need to pluck some fresh green leaves. These are the source of your pigments. Then get a mortar and pestle to grind the leaves. You will also need a pencil to draw the baseline, a suitable solvent, some filter paper, pair of scissors, and some beakers. Select fresh, healthy green leaves for this experiment. The fresher the leaves, the better the pigment extraction will be. So make sure they are not dried or damaged. Next, wash the leaves thoroughly with tap water to remove any dirt, dust, oil, or other contaminants. This step is crucial to make sure nothing interferes with the pigment extraction. After washing, cut the leaves into small pieces. This makes it easier to grind them and helps release the pigments more effectively. Then place the cut leaves into a clean mortar. Make sure your mortar and pestle are completely clean, dry, and free from any particles or residues from previous experiments. This ensures accurate results and prevents contamination. Then add a few drops of acetone to the leaves in the mortar. Remember, acetone acts as the solvent here, so don't use water. The acetone helps break down the leaf tissues and extract the pigments. Grind the mixture well until you get a smooth, fine paste. Next, let's prepare the chromatography paper. Take a thin strip of chromatography paper. Make sure it's clean, dry, and cut neatly for best results. Using a pencil, draw a horizontal line about 2 cm from the bottom of the chromatography paper. We use a pencil, not a pen, marker, or ink because colored inks may contain pigments that could interfere with our results. So make sure to use a carbon pencil for this step. Next, use a plastic pipette or even a fine brush or stick to place a small drop of the pigment extract right on the pencil line. The key here is to make a concentrated spot. So if the spot looks too watery or light, you can let it dry for a few seconds and then add another drop on top. You can also use a dryer gently to speed up the drying and make the spot more concentrated. Now let's prepare the solvent. In a clean beaker, pour the running solvent. A common ratio you can use is 9:1. That's 9 parts ether to 1 part acetone. This solvent mixture helps carry the pigments up the paper during chromatography. Hang the chromatography paper inside the beaker so that the bottom edge just tips into the solvent. Make sure the pigment spot stays above the solvent level as I have shown in the diagram. It shouldn't be submerged. Now let the setup sit undisturbed and allow the solvent to rise up the paper through capillary action. Give it a bit of time. So now when the solvent front is near the top, carefully you have to remove the paper from the beaker and immediately mark the solvent front with a pencil. This step is crucial because you need it later to find out RF value. So now, as you can see in the diagram, when you take the paper out of the beaker, it will look something like this. At the top, you will see the solvent front. Below that, you will notice the pigments have separated at different levels. based on how they interact with the stationary and mobile phases. Hope this video was clear and informative. If you haven't already subscribed to Pearson Bio Academy, please subscribe to stay updated with all our future content. Don't forget to hit the like button if you found this helpful. Thank you for watching and stay tuned for more videos coming soon.